Hello, my name is Samantha, and I will be presenting for myself along with Rachel and Andrew, and today we will be discussing dissolvable biomaterials for wound healing. To start off, we have a brief history of biomaterials for wound healing in general to give some background. As early as 1600 BC, there are records of Greek doctors using catgut, the twisted intestines of sheep and horses, to sew up wounds for gladiators after battle. In AD 150, Egyptians started using sutures more closely related to those used today. In the time between then and the invention of hydrogels, sterilization and mass production techniques were a main focus for sutures. It wasn't until 1960 that hydrogels were first invented. In 1974, the first absorbable sutures were released by a company called Vicryl. Finally, in today's age, dissolvable sutures and hydrogels are a major focus for optimal wound healing. The main motivation and purpose behind having dissolvable biomaterials for wound healing is the elimination of disruption of the wound site. Absorbable sutures eliminate the need to physically remove surgically implanted sutures, and therefore doctors don't need to reoperate once internal tissues are healed. Additionally, dissolvable hydrogels reduce interruptions in the natural inflammatory response, thereby removing secondary trauma induced by mechanical removal. In regards to bioperformance, these dissolvable biomaterials have major implications on the inflammatory response. As we all know, the inflammatory response serves to destroy, dilute, or wall off injurious agents, thereby setting off a series of events that heal and reconstitute damaged tissues. The removal of sutures or hydrogels from wounds can interrupt any of the stages of the inflammatory response, including clotting, provisional matrix deposition, leukocyte signaling, blood vessel formation, collagen deposition, and more generally, repair or regeneration of tissue. For the first article about dissolvable biomaterials, I chose to focus on transparent, conductive, supramolecular hydrogels with stimuli-responsive properties for on-demand dissolvable diabetic foot wound dressings. The objective of this study was to develop a hydrogel that is dissolvable on demand, which aids in the wound healing process. The researchers indirectly posed the hypothesis that in creating a specific chemistry for the hydrogel, the properties can be tailored in such a way that optimizes wound healing and reduces the effects of secondary trauma. The supramolecular hydrogels were made by combining varying concentrations of constituents such as silver nanowires, ethylene glycol, copper 2 chloride, sodium hydroxide, sodium tetraborate, and acrylic acid. The picture on the left shows the chemical makeup of the hydrogel, where the picture on the right shows what a hydrogel looks like. Focus was on the hydrogel's ability to remain transparent, antibacterial, conductive, self-healing, angiogenesis-inducing, and collagen depositing. In general, hydrogels should also aid in the wound healing process by allowing porosity for gaseous exchange, and the molecular component should also allow for retainment of a significant volume of water to maintain moisture at the site of the wound. The following experiments sought to test results on the hydrogels and diabetic foot wounds because of their prevalence and current treatment difficulty in medical applications. There were many methods and tests done in this research because there were so many variables being tested, as stated in the previous slide. Firstly, the supramolecular hydrogels had to be assembled to the chosen concentration of constituents by the researchers. The fabricated hydrogels were then cut into pieces and placed in 6-well plates with antibiotics like penicillin and streptomycin. Experimental L929 cells derived from subcutaneous areolar adipose tissue of mice were proliferated on the hydrogels while control groups were treated with DMEM. A live dead assay was performed in reference to the proliferation on the hydrogel surface to obtain initial data. Laboratory rats were injected with streptozotocin, or STZ, to induce diabetic symptoms in order to test the efficacy of the hydrogels in diabetic wounds. Rat skin was biopsied and implanted with Staph aureus and E. coli bacteria. The experimental group was treated with 200 microliters of hydrogel while the control group was washed with PVS. Qualitative data through digital pictures was obtained and quantitative data of wound area was calculated on days 0, 5, 10, 15, and 20. On days 5 and 15, tissue was collected to perform staining procedures that allowed the researchers to evaluate inflammation, morphology, tissue regeneration, and collagen deposition. Further staining provided results on bacterial growth or diminishment, and immunohistochemistry was utilized to view the neovascularization of regenerated granulation tissue. Aside from tests on the tissues, mechanical, electrical, and light energy tests were done on the hydrogels themselves to evaluate strength, self-healing, conductance, and transparency of the materials. To test the mechanical properties of the hydrogel, strain amplitude sweep tests were done in which the hydrogels were stretched at a fixed angular frequency of 10 radians per second with stretch percentages ranging from 5 to 200%. For the electrical experiments, LED diodes were wired through two separate hydrogels that would be severed to evaluate self-healing and conductance. To assess transparency, light energy was sent through the hydrogels in the form of wavelengths, ranging from 400 to 700 nanometers, to test percent transmittance in reference to the amount of silver nanowires in the particular hydrogel. Finally, a second qualitative transparency test was done by laying pieces of the hydrogel on top of the letters SKL, JLU to assess readability. In comparison to control, there was a significant decrease in wound area, wound severity, bacterial presence, and number of inflammatory cells after 20 days of healing with the supramolecular hydrogel. Additionally, significant increases in percent of collagen deposition and number of blood vessels were seen after application of the hydrogel in comparison to control. The live dead assay illustrated the similarities between the experimental and control groups. That is, there was no significant difference in cell proliferation between the groups, so the hydrogels proved viable. In reference to the mechanical tests, it was found that percentages of 200% stretch did not allow for self-healing of the hydrogels. However, at lesser percentages, the hydrogels quickly bounced back to their original mechanical properties and size. Electrical properties of the hydrogels were also able to be restored after severance. When the hydrogel was cut into two pieces, the LED light was extinguished. However, after 2 hours, the hydrogels were completely repaired and the LED light turned back on. Transparency is a crucial component of hydrogels because of the ability for qualitative review by medical professionals without the necessity for removal of the biomaterial. It was recorded that the average transmittance at 660 nanometers decreased from 93% to 74% when silver nanowire concentration only increased from 0 to 0.2 milligrams per milliliter. In alignment with the diminished transparency with the addition of silver nanowires in the transmittance test, reduced transparency was also seen in the qualitative SKL-JLU test. Figure 1 in the upper left-hand corner shows the difference in wound area for control on the top and hydrogel on the bottom. You can see that there is a large difference in wound area and severity by day 20. Going counterclockwise, figure 2 on the bottom left shows 4 separate graphs obtained through immunohistochemistry and tissue samples. The top left graphic in figure 2 shows the wound residual area over 20 days, which is just a quantitative analysis of figure 1. The bottom left-hand graphic shows the number of inflammatory cells on day 5 and 15. On both days, there was a significant decrease in inflammatory cells for the hydrogel in comparison to control. In the upper right-hand graphic, the percent of collagen deposition is depicted. Again, a significant increase in comparison to control is seen here, with about a 45% collagen deposition by day 20 for the hydrogel. Finally, the bottom right-hand graphic of figure 2 shows the number of blood vessels, which again significantly increased in the case of the hydrogel against control. In figure 3 in the upper right-hand corner is a visual aid to describe the conductivity test. You can see in the middle picture that the orange hydrogel has been severed. In the right-hand picture, the hydrogel was able to heal itself and was conductive once again, and the light turned back on. In figure 4 on the bottom right, both of the transparency tests are shown. Part A on the left is in terms of wavelength. You can see that as the concentration of silver nanowires increases, the percent transmittance of light decreases. On the right, qualitative analysis was done using these letters to test the visibility through the hydrogel. Figure 5 is a visual depiction of the dissolution of the hydrogel from the wound. You can see that as the pH solution is added to the hydrogel, it easily releases without affecting the wound itself. This is a great depiction of the fact that the removal of the hydrogel is not causing any secondary trauma to the wound. Dissolution of the hydrogel can also be done using ultrasound and solutions containing diol-containing molecules. Because of the molecular components, like molecular entanglements and secondary forces, like hydrogen bonds, ionic bonds, or hydrophobic associations, physical interactions can be altered to force dissolution of this particular variation of hydrogel. The limitations in this study is that dissolution of the hydrogel was done, but tests were not done on effects to the wound afterwards to see if any detrimental effects occurred that were not apparent to the naked eye. Additionally, questions remain about the length of time one hydrogel can be used. For example, how long do these hydrogels exhibit antibacterial properties? However, despite these limitations, the hypothesis that changing the chemistry of the hydrogel would allow for properties to be altered and dissolution to be possible was proved valid nonetheless.
The second article is about biocompatibility of dissolved materials, and I personally decided to focus on the accelerated wound healing with extracellular matrices on the surface of surgical sutures. In reading this article, the researchers first pointed out a couple flaws in today's surgical sutures, namely that a lot of them can cause unwanted infections and secondary trauma to the soft and fragile tissues that they're put through. So in order to fix this, the team actually wanted to go in there and add unfolded fibronectin to the surface of polydextrone sutures, or PDO for short. And to do that, they used the following two main methods. So the first method is where they dip the PDO sutures into a bath of polystyrene sulfonate. And this solution was about a 33 molar percent concentration of sulfate groups, and this caused a negative charge on the surface of the PDO sutures. Similarly, the UV ozone and radiation would also cause negative charge on the sutures. However, the way it goes about doing it is you actually put the sutures into a bucket of water and you add ozone into that container. And the ozone will actually react with the water and create an acidic solution. And then when you add UV, it also will go in there and create a negative charge to the suture's surface. And this is really important because it's that negative charge that allows the fibronectin to attach and stretch out over the entirety of the structure. After they were able to get the fibronectin to attach and stretch out, they went in there and they did several tests, but I put a handful of them on the slide for you guys today, and we're going to really go into the first three more in depth, being the adsorbed fibronectin, or how much fibronectin was actually on the surface, the in vitro testing, and the in vivo testing. So for fibronectin absorbance, the polystyrene sulfonate solution actually caused more fibronectin to adsorb to the surface of the sutures. This really makes sense why the polystyrene sulfonate sutures had a larger amount of fibroblast area overall. So to test for the fibroblast area, they went in there and they had a plate full of fibroblasts near 100% confluency. Then they took the back of a spoon and they scratched the surface of it just enough so that way they placed down the suture. And then they left the suture in the dish and in an incubator for 72 hours, and then removed the suture and measured the distance between the two sides of the scratch marks. As you'll see later, it really looks almost like a canyon, and so it's really easy to actually tell which ones do better. And finally, they looked at the scar formation in the in vivo test. The part of this that kind of surprised me was that the polystyrene sulfonate, which had more fibroblast activity and fibronectin adsorbance, wasn't statistically different from the ultraviolet irradiation. And I found that really interesting. So this is going to be the general process in figure 6 of how the fibronectin was attached. As you see, they actually take just a plain suture and they either do their PSS or polystyrene sulfonate dip coating or their ultraviolet ozone irradiation to create a negative charge on the outer surface of the suture. Then after that, they just dip the negatively charged suture into a fibronectin solution, and after it sits for a bit, the negative charges will attract globular fibronectin and slowly stretch it out. Then in figure 7, we can actually see the comparative graphs between how much fibronectin had absorbed to each one of the materials. For this slide, we're looking at a lot of the in vivo testing. So in figure 9, you can really see what I was talking about earlier with the canyon looking structures, where the green is actually the fibroblasts that have been dyed. And in figure 8, they also show just how much growth of cells was present after 24 and 72 hours on the plate after the initial damage had been done. Then in figure 10, it is another comparison, but this one is a more qualitative version of figure 9, where they actually measured the average distances between the two fissure lines and added the standard deviation, and we got figure 10. In figure 11, they went in there and they looked at the amount of collagen area in terms of percent, so that way they could tell how much collagen is actually being deposited by the body with the help of the fibronectin attached to the sutures. And then figure 12 is more of a schematic representing how they actually went in there and created lacerations upon the underbelly of male Baltic mice. And then in figure 13, it shows the average epithelial thickness after the 10-day healing time of each one of the different suture types. Some of the limitations of the study could potentially be fiscal practicality, the fact that they only used one type of adsorbable suture, and how they only really tested superficial wounds on in vivo studies. So the reasons why these are limitations is we don't know if these results would actually go in there and be reproduced on other different absorbable suture types. And so it's relatively solid for this one type of suture material. However, it's not the only type of adsorbable suture. Then, and the other issue is that with in vivo testing, they only really did the top layer. So we don't really know how deep these sutures can go before they undergo some mechanical wearing. Maybe the body will scrape off some of the fibronectin since it is only kept on with negative charges. Last is fiscal practicality, and I do not know how physically practical it is to have large amounts of fibronectin and the PDO sutures along with the polystyrene sulfonate solution.
The third and final study explored the application of static fatigue testing to the behavior of absorbable sutures. The main focus of the study was to develop and validate a new technique to determine how absorbable sutures react to static loads over different time periods. In order to confirm the effectiveness of the newly developed technique, it was expected to reliably measure the time of fracture of dissolvable sutures under the static load conditions that they would be subject to upon implantation in the body. For this study, coated visceral sutures were used as the testing material. The sutures were braided and placed inside of a tube which was contained in a temperature-controlled water jacket that was maintained between 36 and 37 degrees Celsius, which is representative of the human body temperature. A water bath was filled with isotonic saline using a hypodermic needle through a rubber stopper at the bottom of the test environment in order to prevent air pockets. The tube with the suture was then added to the bath. A series of static loads between 4 and 34 newtons were applied to the suture via dead weights, and the times and loads at which the sutures fractured were recorded. The results of the study validated the effectiveness of the developed technique. The data obtained via testing in simulated biological conditions proved to be consistent with the values for suture strength reported by the suture's manufacturer. For instance, at a load of 34 newtons, the tested sutures were found to break almost instantaneously, which was consistent with the reported breaking strength of coated visceral. The strength retention values of the sutures obtained during testing also proved to be consistent with their theoretical values, matching the specifications for strength degradation over time detailed by the manufacturer. Due to the successful nature of the test results, the developed method has the potential to be used in future studies that involve static fatigue testing on absorbable sutures. In addition, the development of this method may provide a concise way for those manufacturing dissolvable sutures to test the effectiveness of potential products when it comes to handling static loads and making appropriate changes. The study had a few limitations that should be addressed. For one thing, only 2 trials were performed before the data was analyzed. This called the validity of the study's results into question, as a larger number of trials would have been needed to confirm their legitimacy. In addition, only a single brand of dissolvable sutures were tested, which left it uncertain whether or not the derived testing method was universally applicable. The testing was also performed in vitro as opposed to in vivo, which left some uncertainty as to whether or not the obtained data was completely reliable, although it should be noted that this limitation is present for all studies performed in vitro. The 3 studies explored throughout the research project had multiple similarities and differences in their experimental methods, motivations, and significant findings when compared to each other. For example, while both studies 1 and 2 were performed both in vivo and in vitro, the third study was only performed in vitro. All 3 studies, however, involved the use of dissolvable biomaterials and aimed to fix biocompatibility issues. Some studies also had aspects that weren't shared with either of the other 2 studies, such as the first study being the only one to have significant findings related to wound severity.
So some of the future ideas for these topics would be new and improved surgical sponges, because surgical sponges do have a tendency to be left in the body after they've soaked in a large amount of blood, because they start looking just like the tissues around them. However, with non-absorbable sponges, they would have to do a corrective surgery and actually remove the sponge, and this can cause even further damage to the site. Another possible use is it limits the amount of frontier medicine, or in other words, people try and go in there and do medical practices at home the best they can. So with regular sutures, a lot of people will actually just try to remove them themselves, and this could cause other sort of complications if they do it improperly, and it would cause a need for more revisions and potentially more sutures. So by having strong dissolvable suture material, they do not need to go in there and actually remove the sutures themselves. Another possible factor of this is deeper internal incisions, since the strongest types of suture material aren't necessarily degradable in the body. And so going in there and learning more about the different stresses that each of the sutures undergo, and learning about how they can be incorporated and absorbed by the body, can actually cause us to have more confidence with deeper internal incisions with biodegradable sutures. Finally, would be really nice in a lot of superficial injuries, even things like plastic surgery, due to the fact that there's less secondary trauma with a lot of these suture types. And this causes noticeably less scarring, especially as we saw in some of the mice in some of the studies.
Thank you for watching our presentation, and here are our questions.